Hello all together and welcome to my poster presentation about exhaled breath as a complementary metrics and doping controls. In the following, I would like to present some data from an administration study with Propranolol. My name is Anne-Marie Garzinsky and currently I'm a PhD student at the German Sport University in Cologne. I would like to start with a short introduction, why there is an interest in research to find complementary matrices to blood and urine that are currently used as matrices for the detection of doping agents. There are several disadvantages, especially concerning the sample collection procedure. Since blood collection is an invasive procedure and the provision of urine is an invasion of the athlete's privacy, with exhaled breath as a complementary matrix, the sampling procedure is less invasive, less intrusive, and less time-consuming. However, there are also advantages from an analytical point of view, for example, concerning the stability of the analytes. In elimination studies conducted to date, it was observed that the detection window for exhaled breath tend to be shorter than in urine or blood. And this suggests an application for in-competition controls. Among the relevant groups on the WADA Prohibited List, narcotics, cannabinoids, and certain stimulants have been thoroughly examined owing to the interest in exhaled breath for drug of abuse testing. In order to investigate the detectability and exhaled breath of beta blockers, an elimination study was performed with Propranolol as a representative that I would like to present in the following. Therefore, 40 milligrams of Propranolol were administered to a total of 19 volunteer participants. Among these participants were male and female non-smokers, as well as male and female smokers. After the intake, 7 exhaled breath samples were collected within the next 24 hours, and at 4 time points, additional dried blood spot samples were collected as a reference matrix. For the preparation of the samples, deuterated propranolol was pivoted onto the electrostatic membrane of the exhaled breath cartridge, and the trapped substances were extracted with methanol using centrifugation. The dried blood spots were extracted with a mixture of methanol and purified water containing formic acid for 30 minutes at 37 degrees. After the evaporation to dryness, the samples were reconstituted and measured using high-resolution mass spectrometry in targeted MS-MS mode. The method was characterized with regard to the limit of detection, limit of identification, precision, linearity, and the selectivity, and propranolol was detected from 5 picogram per cartridge in exhaled breath samples and from 0.5 nanogram per milliliter in DBS samples. This diagram shows the maximum concentrations of propranolol in DBS and in exhaled breath, obtained from male and female non-smokers. In most cases, these maximum values were detected in samples provided 1 or 2 hours after the drug intake. Except for P5, HOCB samples were all tested negative for propranolol. For P6 and for P7, the concentrations were comparably high, which is why here is a zoomed-in view. While the measured propranolol concentration in DBS varies by about 40%, the variations in exhaled breath exceed 200%. But there is no correlation between high concentrated exhaled breath samples and high concentrated DBS samples. And there is also no observable correlation with the gender. The consumption of cigarettes has a severe influence on the constitution of the lungs and thus also on the bronchial lining fluid, and so the question arose whether smoking also affects the detectability of non-volatile substances in exhaled breath. To compare the propranolol concentrations in exhaled breath obtained from non-smokers and smokers, we delete the DBS bars and push together the EB bars. And here are the maximum measured concentrations obtained from smokers. But since this comparatively highly concentrated sample skews the results, here is again a zoomed-in view. The results show that propranolol levels in exhaled breath fluctuate significantly in both populations. So that overall there was no consistent tendency observed for cigarette consumption to influence the detectability of propranolol in exhaled breath. But in addition to the inter-individual differences, variations were also observed with regard to the concentration curve along the collection period for the same participant. After a decrease in the EB concentration after about 4 hours, a new increase was detected after 6 to 8 hours. And this was observed in the case of several participants. These fluctuating propranolol levels in EB could be due to differences in terms of the exhaled particle concentration, so that the amount of aerosol per volume either increases or decreases. In conclusion, propranolol was shown to be generally detectable on exhaled breath, but the measured concentrations were observed to differ both inter-individually and intra-individually. But there was no correlation found with gender or the consumption of cigarettes. The differences are potentially due to fluctuating particle concentrations in the exhaled aerosol. In general, substance amounts in exhaled breath tend to be low in comparison to blood or urine samples, which requires sensitive analytical methods. In one case, the propranolol level may have undercut the limit of detection. The aims for the future are the identification of a normalization factor, for example, an endogenous substance, to elucidate the source of the observed intra- and inter-individual differences, and to relate plasma and exhaled breath levels. To expand the knowledge about the pulmonary elimination of doping substances, further post-administration data are required. Since the sampling procedure of exhaled breath is particularly characterized by its simplicity, it has the potential to address new application areas for doping control. Finally, I would like to thank our sponsors as well as my colleagues from Center for Preventive Doping Research and the Institute of Biochemistry, and of course I would like to thank you for listening to my poster presentation.